Good morning, guys. I hope that you can hear me and that you can see the presentation. And today we discuss the last topic for Colloquium 2. Colloquium 2 is about the tissues of internal environment, and the last type of tissues of internal environment that we will discuss are skeletal tissues, bone tissues, and their development, which is called osteogenesis. So, skeletal tissues include cartilage tissues that we did discuss previous time, and bone tissues that we will discuss now. Bone tissues combine four types of tissues, which are reticulofibrous bone tissue, lamellar bone tissue, dentin, and cementum. Dentin and cementum are the tissues of tooth, and we will discuss these types of bone tissues when we will discuss the structure of teeth. Now we will discuss these first two types of bone tissues, reticular fibrous and lamellar bone. As all other types of tissues of internal environment, bone tissues develop from mesenchyme, which develops from two sources, neural crest and sclerotome. Neural crest gives rise to bone tissues of the face region of embryo, such as bone of jaws, upper and lower jaw, and all other bones of skeleton develop from sclerotome. And here you can see how mesenchyme looks like, which consists of migrating cells of both neural crest and sclerotome. These cells migrate to find their final position to start to develop into skeletal bone tissue. So, as all other tissues, bone tissue consists of cells and extracellular matrix. Cells include three types: osteoblasts, osteocytes, and osteoclasts. Osteoblasts and osteocytes are the cells of the same differome, but osteoblasts are not fully mature cells. They are capable of division, they are active, and they are located upon the surface of the bone. In this picture, you can see a part of bone tissue. This structure, composed of a bone, is called the bone spicule. It is pink in color. It is produced by the cells, and here at the surface you can see the cells osteoblasts. They are located at the surface. Osteocytes are the final form of osteoblasts. They are fully mature cells, and they are embedded into the extracellular matrix of the bone. So osteocytes are located inside the bone. They are not active, and they are not dividing cells. And osteoclasts are the cells of the second differome. They also are located on the surface of the bone. They are multinuclear cells with many nuclei. They break down the bone tissues. Extracellular matrix is composed of some amount of water, about 25%, inorganic substances such as hydroxyapatite crystals, the calcium phosphate salts, and organic substances, about 25%. Organic substances are the fibers, mostly collagen type 1, and some amount of collagens of other types, and amorphous substance or ground substance, which is composed of proteoglycans and glycoproteins. Proteoglycans include proteins and glycosaminoglycans such as hyaluronic acid, chondroitin sulfate, and keratan sulfate. These are the substances that are also present in other types of internal environment, such as fibrous connective tissues and cartilage tissues. Glycoproteins are adhesion proteins for binding components of extracellular matrix together. They include osteonectin and osteopontin. These are the molecules which can help to attach crystals of hydroxyapatite, crystals of calcium phosphate salts, to collagen fibers to make the hard extracellular matrix of the bone. So in bone tissue we have a lot of cells, a lot of living cells capable of breaking down the bone tissue and making the bone tissue again to remodel bone. And these are the cells of two differons, both in embryo developed from mesenchyme. So first differon is the osteocyte differon, which includes pluripotent cell of skeletogenic mesenchyme, which gives rise to osteogenic cell. Next it develops into osteoblast and finally into osteocyte. In our mature body we can find in our bones these three cells: osteogenic cells, osteoblasts and osteocytes. Mesenchyme is not present in adult body, it is present only in embryo during embryogenesis. The second differon which is found in bone tissue is the differon of osteoclast. Actually, osteoclast develops from monocytes of blood, and monocytes develop from hemopoietic stem cell, which is located in red bone marrow. So monocytes come into the region of the bone with blood vessels, which are also present inside the bones. Then they go out from blood capillaries, develop next into macrophages, and then macrophages fuse together. Few macrophages fuse together to form one multinuclear cell, which is called osteoclast. So here you can see the cells of bone tissue. These are two cells of one differon, osteoblast and osteocyte. Osteoblast is the non-mature cell. It can divide and make new cells, and its major function is to produce all components of extracellular matrix. It is located upon the surface of the bone matrix. You can see it in this picture below. The bone itself is pink in color because of collagen fibers, which make the organic matrix upon which the crystals of calcium hydroxyapatite are connected. So this bone is covered from all the sides with osteoblasts. Some are active and some are not non-active osteoblasts. Those which are non-active look like flattened cells kind of lying upon the surface of the bone. They do not produce components of extracellular matrix just now. They just cover the surface of the bone and they are ready to begin to produce the bone. But these, which are active osteoblasts, and as you can see here, they are kind of standing upon the surface of the bone, and they are just now producing the bone matrix components. Here you can see how such cells look like. So this is the surface of the bone. The cell is kind of standing upon this bone, and it produces extracellular matrix. Actually, it produces first organic materials, primarily collagen fibers that you can see here. As the fibroblasts in connective tissue, they do not produce the mature collagen fibers. They produce the pre-collagen molecules, from which next in extracellular space the collagen fibers are produced via the process of self-assembling without the help of the cell itself. So the cell produces the pre-collagen molecules to make the collagen fibers and all other organic components of bone, such as glycoproteins and proteoglycans. All these molecules are actually the protein molecules, and it means that the cell which produces a lot of proteins should have a lot of well-developed rough endoplasmic reticulum. And you can see rough endoplasmic reticulum well developed in the cytoplasm of this cell. And as you remember, well-developed rough endoplasmic reticulum makes the cytoplasm basophilic, and you can see it in the picture where we have the hematoxylin and eosin staining of bone tissue. The osteoblasts, which are active, have basophilic cytoplasm because of well-developed rough endoplasmic reticulum. Also, they have Golgi apparatus and rather large and light in staining nucleus. It indicates that the cell is actively producing a lot of proteins. So first, the cell produces organic substance, which is called osteoid. It consists of collagen fibers and glycoproteins and protoglycans. And during the next step, the osteoid, which is lightly stained, this is this light thin area between the cell itself and pinkish stained mature bone tissue. So this area is the osteoid, non-mineralized yet bone tissue. And next step in producing the extracellular matrix of the bone is the calcification or mineralization of the osteoid. It means that next, osteoblasts start to produce some substances that ensure accumulation of a lot of calcium salts and phosphate salts in the area of the osteoid, and so the crystals of calcium phosphates start to grow. They grow and grow, become larger and larger, and they are attached next to the collagen fibers. And so the osteoid becomes gradually mineralized. And finally, we have the hard extracellular matrix, in which the hardness is due to the calcium salts attached to collagen fibers. And actually, when you see this growing bone spicule, you can actually say into which side it will grow next hour, for example. If you see that our bone is covered with non-active osteoblasts here and active osteoblasts on the other side, you can say that it will grow in this direction, upwards in this picture, because these active osteoblasts will make new layers of bone tissue here. Osteocytes are the final form of osteoblasts. They are fully mature cells, and they have smaller amount of cytoplasm, small dark nucleus, because it's not active in producing the components of extracellular matrix. And the osteocytes are located inside the bone spicule itself, inside the bone. Osteocytes are embedded inside within the hard extracellular matrix. How it happens? It happens during the production of extracellular matrix with the osteoblasts. Osteoblasts produce extracellular matrix around themselves, and they gradually embed themselves inside the bone. And when they are embedded inside the bone, they become osteocytes, and they cannot produce no extracellular matrix anymore. They can just maintain the extracellular matrix. And the hard extracellular matrix is not capable for diffusion of any substances through it, because it's hard. No diffusion can happen from other tissues to the osteocytes, but the osteocytes are located inside the small cavities, these whitish area around the cell, which is seen here. It is the small cavity which is called lacuna, and each osteocyte is located inside these small lacuna, and all the lacunae are connected together with small channels, which are called canaliculi. And these canaliculi lead to the surface of the bone. So all the osteocytes are connected together and to the surrounding tissues. So the diffusion can happen through these small channels inside the hard bone extracellular matrix. And this is how these osteocytes receive their nutrition. Here you can see the processes of osteocytes. Osteocytes are connected together with their processes. They have nexuses on the tips of their processes, and some small molecules can pass from one osteocyte to another one through these small channels, and their processes are located inside the canaliculi, through which the diffusion can pass through the bone to the osteocyte. And this is how osteocytes receive nutrition and oxygen. So all the organic substances which we have in extracellular matrix of the bone tissue are produced by osteoblasts, such as collagen fibers of collagen type 1, protoglycans, and glycoproteins. All these substances are produced by osteoblasts. And the third type of cell, the cell of the second differon, is the osteoclast. Here you can see it. It is also the cell which is pictured on your album, on your first page of your workbook. It is the multinuclei cell because it has a lot of nuclei inside it. It develops from monocytes of blood. Monocytes go out from blood vessels, form macrophages, then macrophages fuse together and finally form osteoclasts. Osteoclasts are located upon the surface of the bone. Here in the picture you can see the bone spicule, a structure of bone tissue. Inside it you can see osteocytes, at the surface osteoblasts, and also upon the surface we can see here very large cell with a lot of cell nuclei, which is called osteoclast. Its major function is to break down extracellular matrix of the bone in the process of bone remodeling, which means rebuilding of the bone. Actually, our bones are constantly rebuilding inside our organism. It means that depending upon the forces which cause the change of shape of our bones, this process is led by the help of osteoclasts that can break down some amount of bone from one side, and the osteoblasts will in turn make some portion of bone from other side. And this happens constantly during all our lifetime, including the bones of our jaws. And as the dentists, you will learn how the orthodontic constructors perform their functions, such as brackets, to make the shape of our teeth more correct. How they act? They actually help to rebuild the bone tissue of jaws. So brackets do not change the shape of teeth, but they help to change the shape of the alveolar bones inside which the teeth are located. So the brackets push into some directions to the alveolar bone, and it becomes rebuilt. It is broken down from one end and is built from other end, and thus it changes its shape. And it can happen during all our lifetime, and it means that you can prescribe the brackets for a person which is kind of 80 or 90 years old, because the process of bone remodeling, bone rebuilding, goes all our lifetime. Now, the structure of osteoclast include the main part of the cell, in which you can see a few nuclei, Golgi apparatus, a lot of mitochondria, some rough endoplasmic reticulum, then the so-called clear zone, which helps to attach osteoclast to the surface of the bone. And in this area, the osteoclasts form focal junctions, which are temporary junctions of the cell to extracellular matrix. And below the area of the clear zone, the osteoclast has such folds of cytoplasm, which are called ruffled border. And below the folds of cytoplasm of ruffled border, we have the zone of resorption. How this zone of resorption is formed? It is connected to the function of the osteoclast to break down the extracellular matrix. How it breaks extracellular matrix? First, it dissolves mineral matter. It means that it first dissolves calcium crystals, and from chemistry you know that calcium crystals are not solved in water, but they can dissolve in the acidic environment, in acids. And so osteoclast, when it sits upon the surface of the bone, it attaches to the bone and starts to produce the H plus ions, protons. H plus ions go out from the cells to the zone of resorption, the space between the cytoplasm of the cell and the extracellular matrix. These protons are produced with the help of the ionic pumps located inside the cell membrane, and this is why the cell has a lot of such folds to increase the surface of the membrane, to incorporate inside this membrane more ion pumps, proton pumps, to pump out proton ions. These proton ions go between the cell and the surface of the bone in this resorption zone, and so the microenvironment here becomes gradually with lower and lower pH level. And in lower pH level, in acidic environment, the calcium crystals become to dissolve. The calcium crystals dissolve to calcium and phosphate ions. These calcium and phosphate ions go to the cytoplasm of the cell, then out from the cell to surrounding tissue and to blood capillary. And this is connected to one of the primary functions of osteoclasts. They are not just help to rebuild our bones, but they help to maintain the proper level of calcium salts in our blood. The level of calcium is very important for our functioning, because calcium is needed for breathing and for heart beating and for nervous system. If the calcium level drops, it will lead to very soon death. So osteoclasts help to maintain the proper level of calcium, because they take the calcium from bone and provide these calcium to blood capillaries. So when we have dissolved the inorganic mineral matter of the extracellular matrix of the bone, then the collagen fibers and all other organic materials can be also broken down, and these are broken down with the help of lysosome enzymes. So the cell next provides the lysosome to release their content, the enzymes, via exocytosis, also in this zone of resorption, and these enzymes break down the collagen fibers and other organic components of the extracellular matrix of the bone. And so, step by step, the hard extracellular matrix become broken down. So a small cavity becomes visible, and when the work of osteoclast is over, the focal junctions break down and osteoclasts go away from this area. Here you can see that this osteoclast has broken down some part of the bone tissue in this area. Some more pictures of these three cells that we can see in bones. Osteoblasts, non-mature cells capable of division, which major function is to produce components of extracellular matrix. Here you can see collagen fibers produced by osteoblasts. They have a lot of rough endoplasmic reticulum. They produce first organic substances, then they provide mineralization of these osteoid, which is the name of organic substances, and they gradually embed themselves inside the bone and become next osteocytes. Osteocytes are located inside the hard bone, which is black in this picture, but they are located in small cavities called lacuna, and their processes are located in small channels, canaliculi. Their processes are connected together with the tight junctions, nexuses. Osteoclasts have a lot of cell nuclei, ruffled zone, resorption zone. It breaks down the hard component of extracellular matrix of the bone. And now we will discuss the types of bone tissues. We have 4 types of bone tissues, and we will discuss first 2 of them, reticulofibrous and lamellar bone tissues. Here you can see the general structure of these bone tissues and their differences. So first of all, reticulofibrous bone tissue is the primary, non-mature tissue, and lamellar bone tissue is the secondary or mature tissue. It means that during development of our bones, during embryogenesis, always reticulofibrous bone tissue forms first, and then during the process of remodeling, rebuilding of the bone, it gradually is substituted with the secondary, mature lamellar bone tissue. And also during rebuilding of the bone in our adult body, especially during the processes of the healing of the breaks, when we break our legs or arms, for example, first reticulofibrous bone tissue is formed, and then it is rebuilt to form lamellar bone tissue. Next, reticulofibrous bone tissue is less hard than lamellar. Lamellar one is more hard. Why so? Because reticulofibrous bone tissue is less mineralized, and lamellar bone tissue is more mineralized if you compare it to reticulofibrous one. More mineralized, it means that it has higher amount of calcium salts than reticulofibrous one. Next, reticulofibrous contains irregularly arranged cells, osteocytes, and extracellular matrix. No any type of arrangement. They are chaotically located inside the reticulofibrous bone tissue. But in lamellar bone, we have very structured organization here. and a lamellar bone consists of functional and structural units, which are called bone lamellae. Lamellae, or single lamella, is the plate of the extracellular matrix, which consists of collagen fibers embedded into the hard inorganic material, and the collagen fibers in one lamella all go parallel to each other, but in adjacent lamellae the angle between the collagen fibers is about 90 degrees. The osteocytes are located between the adjacent lamellae, not inside the lamella but between them. In reticulofibrous bone tissue we have higher content of cells. It means more cells, less extracellular matrix. In lamellar bone tissue we have lesser content of cells. It means lesser osteocytes, more extracellular matrix. And finally, in reticulofibrous bone tissue, the collagen fibers in extracellular matrix, which are striated in this picture, are located chaotically with no order. They go in any direction they want. But in lamellar bone tissue, the collagen fibers are regularly arranged parallel to each other in one lamella, and orthogonal, it means with the 90 degree to each other in adjacent lamellae. First, reticulofibrous bone tissue, so it develops during embryogenesis, and also it is formed where the bone is remodeled, rebuilt. So here you can see that the cells have chaotically arrangement, between them extracellular matrix, in which we have the collagen fibers and some other organic components, and inorganic material. Osteocytes are located inside lacunae, small cavities. Their processes are located in canaliculi, which means small channels. Here you can see the transmissional electron microscopy picture of reticulofibrous bone tissue. You see here some osteocytes, and between them extracellular matrix with collagen fibers, which are cross-striated in transmissional electron microscopy pictures. They go in every direction, no any arrangement of them. And we can see this tissue in our histological preparation of the direct development of the bone from mesenchyme. So here we can see some bone spicules, the structures of the bone tissue. They are pink because of collagen fibers. Inside them we see the osteocytes inside lacunae. The spicules are covered with osteoblasts, and osteoclast is present here. Everything which is between is the mesenchyme, from which this bone tissue develops here in this histological preparation. And also we can see this bone tissue in our second histological preparation of the osteogenesis, which is called indirect osteogenesis. This is the process of development of the long bone, the bone of the leg, for example. So here also first the reticulofibrous bone tissue forms, and during next steps of development of this long bone, the reticulofibrous bone tissue will be rebuilt. It will be broken partly and substituted with the lamellar bone tissue, which is mature bone tissue. So lamellar bone tissue is the mature type of bone tissue. Its structural and functional unit is the bone lamella. Here you can see 3 bone lamellae. Inside every one of them, the collagen fibers go parallel to each other, but in adjacent lamellae, they are orthogonal to each other. It means 90 degree to each other. Osteocytes are located just between the adjacent lamellae. Osteocytes are located in lacunae. Lacunae cavities are connected with the canaliculi or channels inside which the processes of osteocytes go, and every osteocyte is connected to adjacent osteocytes with the nexuses on the tips of their processes, and also their canaliculi make the network piercing all the bone, and through this network of the canaliculi, small channels, the water with solvents such as nutrients and oxygens can pass through the bone to every cell. And this is how osteocytes stay alive inside the hard bone, because they are connected through the small channels, canaliculi, to the surrounding tissues, from which the nutrients and oxygen can pass to the osteocytes. So, in the bone, we have the compact or cortical and spongy or cancellous or trabecular bone matter in the bone as an organ. And both compact and spongy bone matter is made up with the lamellae. So here we can see the lamellae of the compact bone. The part of the compact bone is the osteon, and in osteon the lamellae are concentrically located. They surround the central canal, inside which the blood vessels and nerves pass. So, as in any other type of the lamellar bone tissue, in any other place where we have the lamellar bone tissue, here in the osteon we can see that in adjacent lamellae the collagen fibers go at 90 degrees to each other, and osteocytes are located just between the adjacent lamellae. And here is the cancellous or spongy bone. It consists of trabeculae, these areas of bones, and between the trabeculae we have spaces which are called cavities filled with bone marrow. So here are the bone marrow cavities filled with red bone marrow, inside which the formed elements of blood are formed. So these bone trabeculae are also made of bone lamellae. Here we can see bone lamellae, between which the osteocytes are located, with their processes attaching together and which go inside the canaliculi, which are connected to the bone marrow cavities. So in the long bone we have the epiphysis, the end tips of the bone, and the diaphysis. Diaphysis, if we look at the diaphysis, it is formed by the compact material, which is the most volume of the bone, and closer to the central part we have some amount of spongy bone. It just surrounds the inner space filled with the yellow bone marrow in the adult person. But first, when the bone is just formed during embryogenesis, this cavity is first filled with the red bone marrow, which is gradually substituted with yellow bone marrow, which consists mainly of adipocytes, the fat cells. And if we look at the epiphysis of the bone, we can see that it mainly consists of the spongy bone matter, but also some compact bone matter also is present. Both spongy and compact bone matter are made up with the lamellar bone tissue. The bone is covered from outside with the periosteum. It is the covering consisting of two layers, outer fibrous and inner osteogenic, which contains stem cells for the bone tissue, osteogenic cells. And the articular surface of the long bone is covered with the articular cartilage, which is hyaline cartilage. So here you can see the histological structure of the bone as an organ. This is the structure of the diaphysis of the long bone. From outside, it is covered with the periosteum, which consists of two layers: outer layer, fibrous layer of periosteum, which consists of dense, fibrous, irregular connective tissue, and inner layer of periosteum. It is very thin. It contains osteogenic cells and some blood capillaries which give nutrition to the inner layers of the bone. From inside, the bone is covered with endosteum. Endosteum, like periosteum, contains the stem cells, osteogenic cells. Next, just under the periosteum, we have the layer of the outer circumferential lamellae. Outer circumferential lamellae. Circumferential means going around the bone, like this, circumferential lamellae. Here you can see in this picture 3 circumferential lamellae with osteocytes embedded between them. Inside every of these circumferential lamellae, the collagen fibers go parallel to each other, but in adjacent lamellae they are orthogonal to each other. And the osteocytes in these lamellae receive their nutrition from the blood capillaries of the periosteum. And these osteocytes develop from the osteogenic cells of the periosteum. The periosteum is connected to the layer of the outer circumferential lamellae with the help of the so-called Sharpey's fibers. Sharpey is the name of the scientists who invented them. These Sharpey fibers are actually the collagen fibers, one end of which is located inside the outer fibrous layer of the periosteum in dense fibrous irregular connective tissue, and the other end is embedded inside the bone tissue of the outer circumferential lamellae here. And these Sharpey fibers make the outer fibrous layer of periosteum very tight attached to the bone. Also, from outside, from the periosteum, blood vessels come into the bone. Large blood vessels come inside the bone through the Volkmann's canals. Volkmann is the name of the scientist. Canal means channel. And then these channels and the blood vessels inside them branch to form smaller blood vessels which go parallel to the bone inside the osteons here, and these are the Haversian's canals. Havers is the name of the scientist. Haversian canal is the central canal of the osteon. So just under the layer of the outer circumferential lamellae, we have the layer of osteons. Layer of osteons contains osteons themselves. One of them you can see here. It consists of central canal, Haversian canal, inside which the blood capillary and nerves go, surrounding with loose fibrous connective tissue, and circumferential lamellae, bone lamellae of the osteons. And between the osteons we have the so-called interstitial lamellae. These are the remnants of the previously formed osteons. As I said before, the bone tissue is constantly partly broken down with osteoclasts and partly built up again with osteoblasts, which means rebuild constantly, and so we have the remnants of the previously formed osteons, which were partly broken down, and we see them in the form of the interstitial lamellae between the osteons here. And the innermost layer of the bone is the inner circumferential lamellae layer. It consists, like outer circumferential lamellae layer, of bone lamellae which surround the sponge bone matter of the bone, and they also consist of lamellae. Between them we see osteocytes. These osteocytes receive blood supply from the blood capillaries of the spongy bone matter. So here in the section of the bone we have the compact bone, which includes these three areas: area of the outer circumferential lamellae, area of the osteons, and the zone of the intercircumferential lamellae. These three make the compact bone matter of the bone. And here, the inner, closer to the inner space, we have the sponge bone matter, which consists of the trabeculae of the cancellous bone. These trabeculae are those shown in this picture. They also consist of bone lamellae with osteocytes between them, and between the trabeculae we have the cavities filled with bone marrow. It is located here. Small amount of the spongy bone and the large central cavity filled with the yellow bone marrow in adults. So here you can see the part of the layer of osteons, some osteons here. Each osteon consists of central canal, inside which we will have the blood capillary and the nerve, and some lamellae which surround the central canal. Between lamellae we have osteocytes. Osteocytes located inside lacunae. Lacuna is the small cavity inside which the osteocyte is located. Lacunae are connected together with small channels. Inside these channels the processes of osteocytes go, and so you can trace all the canals. They are connected together, and the cells, osteocytes, are connected together, and they all the osteocytes, which you can see here, receive their nutrition from the blood capillaries of the central canal via diffusion through these small channels, like this. Also, the central canal contains the osteogenic cells, which cover the inner surface of the bone. So osteogenic cells will be located here, just around the central canal. If it is needed, from the blood vessel of the central canal, the monocytes can go out. They will make macrophages, then they will fuse and make osteoclasts, which can break down the bone tissue of the osteon from inside, like this, break down the bone. And then from the same blood vessels are surrounded with loose fibrous connective tissue with adventitial cells, and adventitial cells can give rise to osteogenic cells, and then osteogenic cells will develop into osteoblasts, which will make new bone tissue, and this is how the osteons can be rebuilt. So as you can see here, the osteocytes are interconnected to each other with the nexuses on the tips of their processes. So osteocytes receive their nutrition from central canal, and all of them have developed from osteogenic cells, which came also from the central canal. The outer limiting line of each ostone is called cementing line. It is less mineralized extracellular matrix than the extracellular matrix of the lamella. Between the adjacent ostones, we have the interstitial lamellae, which are the parts of non-fully broken previously formed ostones. So here you can see three locations of osteogenic cells in the bone. First location of osteogenic cell. Osteogenic cell is the stem cell for the bone tissue cells. So first location is the inner layer of the periosteum. Periosteum is the outer covering of the bone. It consists of outer fibrous layer and small thin inner osteogenic cell layer. It is shown with this arrow here. So in this osteogenic layer, we have osteogenic cells which can develop into osteoblasts, which start to produce extracellular matrix and develop into osteocytes, and thus they make outer circumferential lamellae. So outer circumferential lamellae develop from the osteogenic cells of the inner osteogenic layer of periosteum. Second location of the osteogenic cells is the central or Haversian canal, which is located in the center of each ostone here. So here we have the osteogenic cells, which also can develop into osteoblasts, osteocytes, and so they form concentric lamellae of the ostones. You can see here how this happens. First is the outermost lamellae are formed with the help of osteoblasts, which produce extracellular matrix, embed themselves, and make next lamellae, which are closer and closer to the central canal here. So in the ostone, the outer lamella was formed, next this one, next this one, next this one, and this one, which is the closest to the center, is the most young formed lamella here. And the third location of the osteogenic cells is the endosteum. Endosteum covers the inner surface of the bone tissue in the bone. It is one layer of cells which contains osteogenic cells, which also develop into osteoblasts and osteocytes and form inner circumferential lamellae. Here you can see the spongy matter of the bone. So, lamellar bone tissue is seen in our histological preparation, which is the cross-section of the diaphysis of the bone with specific staining, which is called Schmorl staining. Schmorl is the name of the scientist. Here you can see at high magnification the outer part of the section. This is the periosteum. It consists of dense, fibrous, irregular connective tissue. And here should be the small, thin osteogenic layer. This is the area of the outer circumferential lamellae, which go parallel to the surface of the bone. They all go parallel. And next, deeper into the bone, we have the layer of ostones. Ostones are rounded in shape here, with central canals in their centers. Here you can see some lighter spots, like small lighter dots. These are lacunae, small cavities, inside which the osteocytes are located. And so you can see that the bone actually has a lot of cells, living cells inside it, and also a lot of blood vessels and nerves. And this is why if you break somehow your leg or arm, it will be very painful and bleedy. At a higher magnification, we see here some part of the periosteum, which is dense, fibrous, irregular connective tissue. from periosteum, the blood vessels go into the bone through the Volkmann's channels. This is the Volkmann channel, inside which the blood vessel should be located. And this is the area of the outer circumferential lamellae. One lamella is here, second lamella is here, third lamella is here. So if you are asked to show one lamella, you should find adjacent osteocytes, like here, and the area of extracellular matrix between them will be one lamella. Osteocytes are located between lamellae, inside the cavities called lacunae. Here you can see lacuna, but no osteocyte is inside it. It was not included in this section. But here you can see the darkly stained body of the osteocyte. It is located inside lacuna, and from osteocyte its processes go into all directions, and the processes of this one osteocyte are connected to the processes of another one here, and the processes of this osteocyte are connected to the surface of the bone, and they receive nutrition from the blood capillaries or the periosteum. And you can trace the processes leading to all the osteocytes. Now here you can see the inner part of the bone in this histological preparation. This is the zone of osteons. You can see the osteons here. Between them the intermediate lamellae, the previously formed non-fully broken osteons. Cementing line surrounds the osteon from outside. Inside osteon we have Haversian canal, filled with loose fibrous connective tissue with blood vessels and nerves. At high magnification, the osteon, this is the border of the osteon, the cementing line. Intercalated lamellae are here. Haversian canal, blood vessel, inside it with some erythrocytes, and lamellae which surround the central canal. Also, you can see here the cavities, lacunae, with osteocytes. One osteocyte you can see here, another one here, etc. Another example of the layer of the outer circumferential lamellae at the most high magnification. Here you can see just lacunae, but here lacunae with the osteocyte itself inside it, the cell body of the osteocytes, and their processes which are located inside small canaliculi, small channels inside the hard bone matter. One lamella is here. It is the part of the extracellular matrix just between the adjacent osteocytes. Another one is here, another one lamella is here, another one is here. So this picture will help you to train yourself to find the correct names for all the parts of the bone of the bone as the organ. And now we go to the development of the bone, which is called osteogenesis. Osteo stands for bone, genesis means development. So bones can develop by two ways: direct and indirect. Direct is also called intramembranous bone formation, and indirect is also called the formation by replacement of the cartilaginous model. The direct means that the bone here develops directly from mesenchyme. Indirect means that from mesenchyme first the model of the future bone is formed from the cartilage tissue, and then the cartilage tissue is gradually replaced with the bone tissue. The direct type of development of the bone is characteristic for the flat bones development, and the example is the development of the jaws. And we will see this type of development in the histological preparation of the frontal section of the jaws of the pig embryo. This process, the direct embryogenesis, starts earlier than the indirect, starting from the second month of the embryogenesis, and indirect development of the bone starts much later, and it is characteristic for the long bones development, such as the bones of the legs and arms. And here you can see the major stages of the both types of development. So indirect osteogenesis directly from mesenchyme. First, the first stage is the formation of osteogenic islet. It means the mesenchyme cells develop into osteogenic cells, which come close together and form some group of cells, which is called islet. Then the cells, osteogenic cells, differentiate into osteoblasts, which start to produce the osteoid. Then this osteoid is calcified, mineralized, and thus the primary reticulofibrous bone is formed. And step 4 is the rebuilding of the bone. It means the primary reticulofibrous bone is broken and is substituted by the secondary lamellar bone. And thus we have the formation of the flat bones. In turn, in indirect osteogenesis, first step is the formation of the model of the bone. It means that mesenchyme gives rise to the cartilage tissue, which forms the shape of the future bone. It looks like the small bone, but it consists not of bone tissue but of cartilage tissue developed from mesenchyme. Second step is the perichondral ossification, the formation of the bone which covers the cartilage tissue in the diaphysis of the future bone. Stage 3 is the endochondral ossification in the diaphysis, formation of the primary ossification centers, and step 4 is the formation of the secondary ossification center in epiphysis. For some time, the area of the cartilage of cartilage tissue stays unchanged between the diaphysis and the epiphysis, and forms the metaphysis or metaphyseal plate, which helps the bone to grow in length. And finally, the last cartilage tissue is replaced with bone tissue, and we have finally the formed long bone. And now we will discuss these processes in details. We start with the direct osteogenesis, which is more simple processes. It starts from the 4th week of development. It means that it starts from the 2nd month of development. It starts with the formation of osteogenic islet. Mesenchyme cells, which you can see here, they have processes, they are migrating cells. They start to differentiate into osteogenic cells and further into osteoblasts. And this happens in the area where we have a lot of blood vessels, because these cells of mesenchyme are actually pluripotent cells. Pluri means many, potencies means abilities, potential ways of differentiation, and the way of differentiation depends on the level of oxygen in surrounding tissue. If the level of oxygen is lower, then the mesenchyme will develop into cartilage tissue. If the oxygen level is higher, then mesenchyme will develop into bone tissue. The level of oxygen is higher in the areas where we have a higher amount of blood vessels. So in this picture we have a lot of blood vessels. So here in this area of the embryo we will have higher level of oxygen, and this is why these mesenchyme cells develop into osteogenic cells and further into osteoblasts. Next, the osteoblasts start to produce the extracellular matrix, pink in this picture, and gradually embed themselves inside it. And the osteoblasts become osteocytes embedded inside the bone spicule. Other mesenchyme cells also develop into osteoblasts, which cover the surface of the bone spicule and continue to produce the extracellular matrix. And this is how the bone grows. First, they produce organic substances, and then organic substances are mineralized. It means that the calcium salts, which come from the blood capillaries, start to form crystals which are attached to the collagen fibers. And thus we have calcification of bone matrix. And finally, not shown here, the last step 4 is the remodeling of bone. It means that reticular fibrous bone is gradually destroyed and replaced by newly formed lamellar bone with the help of osteoclasts, which break down the portion of the reticular fibrous bone, and the osteoblasts will start to form in this area lamellar bone. This is the picture of what we see in histological preparations. So this is the osteogenic islet, which means small island, where the mesenchyme cells, which are seen here, mesenchyme cells have long processes and small cells with small cell nuclei. Here in this area they lose their processes and become osteogenic cells that develop next into osteoblasts. And these osteoblasts develop basophilic cytoplasm because of rough endoplasmic reticulum needed to produce proteins, and they start to produce organic material of the future bone. Next stage we see here, this is the bone spicule, inside which we have already the osteocytes, which are embedded inside the bone. They are located inside the cavities, lacunae. The bone spicule is covered with the osteoblasts and osteoclasts in this area. These osteoblasts are non-active because they are flattened upon the surface, and these osteoblasts are active. They are producing the components of extracellular matrix just now. This area, lightly stained area, is the non-mineralized material, which is called osteoid. Osteoid is the newly formed ground substance of the future matrix. In next stage it will be mineralized. Osteoclast here is the multinuclear cell. It contains a lot of cell nuclei, and it breaks down the bone tissue in this area. So looking at this bone spicule, we can say into which direction it will grow. It will grow to this direction because here the extracellular matrix will be formed with the active osteoblasts, and it will become thinner from this area because here osteoclast breaks down partly the bone. Everything that surrounds the bone spicules is the mesenchyme, small cells with processes. And in this area we have a lot of blood capillaries, which give higher level of oxygen. This is our real histological preparation. This is how it really looks like. So this area is the bone tissue. This is the bone spicule. It is pink in color because of collagen fibers seen here. We see here some embedded osteocytes, and in this area also osteocytes. We can see their cell nuclei. Bone spicule is covered with osteoblasts, which are flattened, non-active in this area, and in this area also, and large and kind of standing in this area. Here we have active osteoblasts. Also, we can see here the osteoclast. It is the large cell with a lot of cell nuclei. Everything which surrounds the bone spicule is the mesenchyme, and also here we have some blood capillaries with erythrocytes inside them. In this area we can see the osteogenic islet, which consists of newly formed osteoblasts. They are all active here. They start to produce osteoid, the pinkish substance here. The basophilic cytoplasm of osteoblasts is due to a lot of rough endoplasmic reticulum needed to produce a lot of proteins of the osteoid. Another example of this preparation, this is the bone spicule, pink in color because of collagen fibers, some osteocytes embedded inside it, osteoblasts non-active in this area, and active ones in this area. Where we have active osteoblasts, it means that here they produce extracellular matrix, and this non-stained area just between the cells and the bone spicule, this non-stained area is the osteoid. All that surrounds our bone spicule here is the mesenchyme, and this is the blood capillary. This one, this, and this. Another example of the same preparation, bone spicule with embedded osteocytes. Osteoblasts cover the surface, non-active flattened, active standing with a lot of cytoplasm, and here the osteoclast. Mesenchyme surrounds the bone spicules. This is how this histological preparation looks like at lower magnification. This is the epithelium of the oral cavity, because this is the part of the frontal section of the lower jaw of the pig embryo. This is the developing tooth of the pig embryo of its lower jaw. We need this area where we see the pink in color bone trabeculae or bone spiculae. They are all covered with osteoblasts, and in some areas we should look for osteoclasts, which are larger. Everything which surrounds it, small cells with processes at white background, this is the mesenchyme. The same at high magnification, bone spicules, osteocytes inside, osteoblasts outside, osteoclast, mesenchyme with blood capillaries. The same here, bone spicule, osteocytes in lacunae, osteoblasts at the surface, large osteoclast with several cell nuclei, mesenchyme. Osteoclast with 1, 2, 3, 4, 5 cell nuclei. Osteoblasts cover the bone spicule here. Osteocytes embedded inside the bone. They are inside the lacunae. Mesenchyme. Bone spicule, osteocyte, osteoblast, mesenchyme cell. Osteoclast here. And now we go to the indirect osteogenesis. It starts with the 8th week of development. It means that it starts from the 8th week is the end of the 2 months of development of the embryo. And this is the way how the long bones are formed. So this process starts with the formation of the model of the future bone, which consists of hyaline cartilage tissue. It develops from mesenchyme. Next, the perichondral bone is formed just below the perichondrium, which covers the cartilage, And this bone, which is called bone collar, blocks the diffusion for the nutrients from the perichondrium to the cartilage cells, and this causes death of cartilage in this area because these cells cannot receive nutrition anymore from the perichondrium because of hard bone of the bone collar. The way how the cartilage dies is very interesting. The cells become larger in size, they become hypertrophic, and the extracellular matrix of the cartilage becomes calcified. It becomes hardened. Then osteoclasts, which go from blood capillaries, break down some areas of the bone collar and then the calcified cartilage and make the way for blood vessels to grow inside the bone, future bone in this area. This is how the inner cavity is formed. It will be the future marrow cavity, bone marrow cavity. It is filled with blood vessels which grow here from outside, and the mesenchyme which goes along with blood vessels. The cartilage continue to be broken down to the epiphyseal area here, and it is partly broken, and upon the remnants, remaining areas of the calcified cartilage, the newly formed bone tissue starts to grow, and this will be called endochondral bone. Next, the secondary center of the calcification forms inside the epiphysis, and it starts to grow in all directions. Next, the cartilage is replaced with bone tissue in all the diaphysis area of the bone. Almost all epiphysis also is replaced with the bone tissue, and only some amount of cartilage tissue remains between the epiphysis and diaphysis. In this area, cartilage starts to develop, start to divide cells of cartilage, chondrocytes, divide via mitosis and form such columns of cells, and these cells form the growing metaphyseal plate, which ensures the growth of the bone in length up to 20-25 years of old. And now we will discuss all these stages one by one. So first of all, the skeletogenic mesenchyme gives rise to the hyaline cartilage tissue and forms the model of the future bone. So all this is the cartilage. Hyaline cartilage, it is covered with the perichondrium, which consists of two layers, outer fibrous and inner chondrogenic layer. In chondrogenic layer, in inner layer of perichondrium, we have actually bipotential cells. They can differentiate into chondrogenic cells if the oxygen level is low, but they can differentiate into osteogenic cells if the level of oxygen is high. Now, during our first stage of development, we have low level of oxygen in this area because we can see no blood vessels here. And this is why our bipotential cells give rise to more and more chondrocytes, which make this future bone to grow, become larger and larger. Now it consists only of cartilage, covered with perichondrium. Next, in this area of developing embryo, a lot of blood vessels start to grow from other areas, and it causes oxygen level to increase in this area surrounding our future bone. And this causes our bipotential cells of the inner layer of the perichondrium to differentiate into osteogenic cells instead of chondrogenic cells. And what will happen? These bipotential cells become osteogenic cells, and they start to form bone tissue here, just below the perichondrium, in the area of the future diaphysis of our bone, here and here. And this bone surrounds the um, future bone in the area of the diaphysis, and it is called perichondral bone. Peri means surrounding, chondral means um, cartilage. So bone collar surrounds the bone, um, our future bone. Now it consists of cartilage tissue. What happens? Our cartilage received nutrition showed with the bluish arrows from the small blood capillaries of the perichondrium via diffusion. But in this area where we now have the bone collar, which consists of hard bone tissue, the diffusion now is not possible because nothing can diffuse through the hard bone tissue. So the cartilage in this area, just below the bone collar in the diaphyseal region of the future bone, start to die, start to degenerate. And how it degenerates? The cells become hypertrophic. The cells, it means that they become larger, they become filled with water, and extracellular matrix calcifies. It means that extracellular matrix in our cartilage becomes hard, and it also blocks the passages of all the nutrients and oxygen. So in this area, the cartilage degenerates. Next, the blood vessels which surround our future bone become very multiple in this area. From blood capillaries, the monocytes go out and they develop into osteoclasts. Osteoclasts make the holes in the bone collar because they can break down the hard component of the hard tissues, such as bone tissue and calcified cartilage also. So osteoclasts make holes both in the bone collar and in the calcified cartilage inside. And through these holes, the blood vessels can grow inside the bone. So inside the bone, in the region of the diaphysis, now we have the cavity made up with osteoclasts and the blood vessels surrounded with mesenchyme, which grow from outside into this cavity. The area of the calcified cartilage is here, and in this region also. In epiphyseal region, at this stage we have just the normal cartilage tissue, pinkish in this picture. Next, what happens next? The calcified cartilage continues to be broken down with the help of osteoclasts, and on the remnants of the calcified cartilage, which looks basophilic, the newly formed bone tissue starts to be grown with the help of the osteogenic cells, which develop from mesenchyme, which goes into this cavity with the blood vessels. So mesenchyme, which is now in the cavity inside the bone, develops into osteoblasts that start to make bone tissue just upon the remnants of the non-fully broken calcified cartilage, and thus the endochondral bone is formed. Perichondral bone continue to grow to the areas of epiphysis. The perichondral bone is formed from the osteoblasts, which develop from the bipotential cells of the periosteum. So this is periosteum, outer fibrous layer, inner osteogenic layer, and here osteoblasts make the bone tissue of the perichondral bone. So, unlike endochondral bone, endo means inside, peri means surrounding. If we look to our histological preparations, the perichondral bone, unlike endochondral bone, consists only of pinkish stained bone tissue, but endochondral bone consists of both calcified cartilage tissue in the center and bone tissue which covers these remnants of calcified cartilage. Next, blood vessels grow into epiphysis area of the future bone also, and here the secondary center of ossification is formed, just like the same way as it is formed previously in the diaphysis region of the bone. And the process of the ossification, the replacement of the cartilage with bone tissue, continues both in direction of the diaphysis to epiphysis and inside epiphysis also. And only between the epiphysis and diaphysis some region of the cartilage still remains. In this region, which is called metaphyseal growth plate, or it is sometimes called epiphyseal growth plate, which is the same. It is the area of the dividing chondrocytes. They divide, and thus they provide the growth of the bone in length. But finally, these cells are also being replaced with the bone tissue, and this stops the growth of the bone in length, and this happens by the 20-25 years of old. So in our histological preparation, we see the bone at the this stage, when we have only primary ossification center, but the epiphysis is it consists here only of cartilage tissue. No secondary ossification center is seen. So during this stage, we have in epiphysis the zone of reserve cartilage. It is normal, non-touched with the process of ossification, just the chondrocytes. Everything is okay with them. If we go from epiphysis to diaphysis, we have in this area zone of proliferation of cartilage, where the chondrocytes start to divide and they form such columns of cells. Next goes the zone of hypertrophy. Hyper means higher than normal. So here the cells become larger, like you see them here. Normal cells are seen here. They are small, located inside lacunae. And in the area of hypertrophy, they become larger. This is the sign of their death, because they become filled with water. Next, we can see here that the extracellular matrix gradually becomes more darker basophilic. This is the sign of the calcification of the extracellular matrix of the cartilage. And next goes the zone of resorption of cartilage. Resorption means the breaking down of something. In this area, the calcified cartilage is broken down with the help of the osteoclasts, which come from the primary bone marrow cavity, which fills now the inner space of the diaphysis region of our developing bone. The bone is covered with the outer covering, which is partly called the periosteum in this region, but in this region it is called perichondrium. Why so? Because in this region, just below our perichondrium, we have the cartilage tissue. And you know that the cartilage as the organ is covered with perichondrium. Chondrium stands for chondroblasts, the cells of cartilage tissue. But in this region, just below our periosteum, we have the bone tissue. This red in color, this is the bone tissue of the perichondral bone. This is the bone collar, which caused the death of the cartilage in the diaphyseal region of the future bone and caused the appearance of the primary bone marrow. So in the area where we have the bone collar, the outer covering will be called periosteum. In the region where we have no bone collar, no bone tissue here, just the cartilage tissue, this covering will be called perichondrium. So here in diaphysis we have the primary bone marrow cavity filled with mesenchyme, which develops into primary bone marrow. These are remnants of non-fully broken down calcified cartilage. Calcified cartilage is broken down with the help of osteoclasts, which are shown here. These are multinuclei cells which can break down the hard tissues. They have broken down some areas of the perichondral bone, here for example, which gave the ability for blood capillaries to grow inside the bone from outside in this area. So we can see the blood vessels inside the bone here. And the osteoclasts continue to eat, to break down the calcified cartilage. And next, newly formed osteogenic cells, which develop from mesenchyme, which covers our primary bone marrow cavity. These osteogenic cells will make osteoblasts, which will start to make the bone tissue just around the remnants of the non-fully degraded calcified cartilage. And thus we will have the endochondral bone. So in this picture we can see at high magnification the area of the metaphyseal plate. In the upper region we have the zone of reserve cartilage, just the hyaline cartilage, it is okay, everything with it. Lower we have the zone of proliferation. It consists of the dividing cells, which form such columns of cells. Lower we have the zone of hypertrophy, where chondrocytes become larger because they become filled with water. It's the sign of their death and degradation. Here the zone of calcified cartilage because of the calcification of extracellular matrix. And next the zone of resorption and ossification. Resorption means breaking down of cartilage. Ossification means the formation of the bone tissue just around the remnants of the calcified cartilage. At high magnification we can see here in this picture the zone of the resorption and the ossification of the cartilage. So here we can see the hypertrophied chondrocytes. They are larger than normal. Lower we have the calcification of the extracellular matrix of the cartilage. Compare how it looks like in this area. Normal extracellular matrix is lighter, and calcified extracellular matrix is darker basophilic in this area. In this region we see that our calcified cartilage is already partly broken with the help of osteoclasts, and we can see here these holes inside our cartilage. These holes are bone marrow cavities filled now with mesenchyme and some blood vessels which grow here. And in this area you can see that just around the remnants of the basophilically stained calcified cartilage, the bone tissue starts to be formed. Bone tissue is pink in color. It is eosinophilic because of the collagen fibers. This bone tissue is formed with the help of osteoblasts. You can see them here. They develop from mesenchyme, which you can see here. Another example of the same preparation. This is the periosteum. Its outer fibrous layer, inner osteogenic layer. In this area it is called periosteum, but in this area it will be called perichondrium, because here no bone tissue is seen, only cartilage tissue. It is the area of the epiphysis. And in this area we have the periosteum, because just below it, beneath it, we have the bone tissue of the perichondral bone, or it is also called the bone collar, which is covering the diaphyseal region of the bone. Our perichondral bone develops from the osteogenic cells of periosteum. You can see here the active osteoblasts, and inside this perichondral bone the osteocytes are embedded inside it, and our perichondral bone consists only of bone tissue. It is equally pink in color. If we go deeper, here we have the cavity filled with the developing red bone marrow and some blood vessels. And in this region we have the zone of ossification and resorption of calcified cartilage. The chondrocytes here are hypertrophied, larger than normal. Extracellular matrix become calcified, more dark basophilic, and here you see that it is partly broken down, and the cavities are filled with mesenchyme and blood capillaries, and already some bone tissue is made up just surrounding the remnants of the calcified cartilage. So here we have the endochondral bone. You should be able to distinguish in histological preparation between the endochondral bone and perichondral bone. Perichondral bone is located just beneath the periosteum, and it consists only of bone tissue. Endochondral bone is located inside the bone, and it consists of two tissues, the calcified cartilage inside and bone tissue outside. The same is in this picture, and this is our histological preparation. This is the process of development of the long bone of the leg of the embryo. This is the epiphysis of the bone, this is the diaphysis, and this is the metaphyseal plate, inside which you can see the columns of the dividing chondrocytes, which ensure the growth of the bone in length. Here we have the primary center of ossification. It means that in the area of diaphysis we have the breaking down of the cartilage tissue and replacement of it with the bone tissue. Bone tissue is oxyphilic in color. This is the periosteum, and this is the bone collar on both sides. So what we see here, in epiphysis we have non-touched, just normal reserve hyaline cartilage tissue. This is the articular surface of the hyaline. It is not covered with perichondrium. In this area our hyaline cartilage tissue is covered with perichondrium. Here it is. It consists of dense fibrous irregular connective tissue and loose fibrous connective tissue of two layers of perichondrium. In this area it is no longer called perichondrium but periosteum, because just beneath it we have already developed bone tissue of the bone collar. This is the perichondral bone in this and in this region. It is formed from the reticular fibrous bone tissue. Inside the diaphyseal region of the bone we have the primary bone cavity filled with mesenchyme, blood vessels and developing red bone marrow. Red bone marrow consists of two tissues, myeloid and reticular tissue. Myeloid tissue is the hemopoietic tissue, which consists of the hemopoietic stem cells and all the cells which develop from hemopoietic stem cells. I mean all the difference of developing formed elements of blood. They comprise the myeloid tissue. Reticular tissue, as you remember, is the stroma tissue. It is the excessive tissue of the red bone marrow, and also some amount of loose fibrous connective tissue also develops here from mesenchyme. At high magnification, you can see here the metaphyseal plate. This is the zone of the reserve cartilage. This is the zone of the proliferation, where we have the columns of chondrocytes. This is the area of hypertrophy, because the cells become larger than normal, chondrocytes become larger than normal. And this is the area of calcification and resorption of the cartilage. This is the perichondrium. It is continuous with periosteum. Periosteum is here in the area where we have the bone color. It consists of bone tissue. It is pink in color here. This is the bone color, perichondral bone. This is the inner cavity filled with blood capillaries, filled with erythrocytes, so they are red in color because of erythrocytes. Between them we have the mesenchyme, which develops into future red bone marrow, and also some remnants of the calcified cartilage covered with newly formed bone tissue. So these are the bone spicules of endochondral bone. The same is here. Perichondrium, periosteum, bone color of perichondral bone. In this area where we have the perichondral bone, just below it the cartilage starts to die. In this area we have the hypertrophied cartilage cells, chondrocytes. They become hypertrophied because they start to die, and they start to die because the bone tissue of the bone color prevent the passage of the oxygen and nutrients from the periosteum. So the chondrocytes cannot receive nutrition and oxygen anymore via diffusion, and this causes their death. In this area the chondrocytes are dividing. They feel good enough because in this area we have no bone tissue between the perichondrium and the chondrocytes, and they can receive nutrition and oxygen from the perichondrium. And this is the zone of normal reserve cartilage. In this area we have the perichondrium, periosteum, endochondral bone. It consists of remnants of cartilage tissue in the center and the bone tissue surrounding it. At high magnification, the zone of hypertrophied cartilage cells, chondrocytes, zone of calcification of the cartilage, where extracellular matrix becomes calcificated and looking more darkly stained, and this is the zone of resorption and ossification. It is broken down with the formation of cavities, which are filled with mesenchyme and blood vessels, and in this area you can see that the osteoblasts start to form the bone tissue surrounding the remnants of the calcified cartilage. This area is the perichondral bone of bone color. It is the perichondral bone because it consists only of bone tissue. No remnants of calcified calcium is seen here, so we can say that this is the perichondral, not endochondral bone. And these are the hypertrophied chondrocytes. They feel bad because the perichondral bone prevents passage of oxygen and nutrients to these cells, and so they start to die. And in this area, the calcified cartilage is broken down, and the cavities are filled with blood vessels and mesenchyme. In this area, you can see the endochondral bones. They have two colors. In the middle area, more basophilic in color, calcified cartilage, and at the periphery, more oxyphilic bone tissue. It is made up with the osteoblasts, which are active in this area. They have large basophilic cytoplasm, and they are kind of standing upon the bone spicule. And in this area, we have the non-active osteoblasts. They are covering the surface. They are kind of lying upon the surface of the bone. In this area, we have the mesenchyme and some developing area of red bone marrow cells. In this area, we have the periosteum, perichondral bone. It is perichondral because it consists only of pinkish colored bone tissue, no remnants of cartilage inside it. And this is the area of hypertrophy, calcification, and resorption of the cartilage. During the bone repair, if you have broken your leg or arm, for example, during the fracture healing, first the bone is resorbed with the help of osteoclasts, which develop from the precursor cells, monoblasts. They break the damaged tissue, and then they go out from this area and make the way for osteoblasts. Osteoblasts develop from the osteogenic cells or from the adventitial cells. They come to this area and start to produce new bone tissue. First they form osteoid, which consists of organic material, and then osteoid is mineralized, and the bone is rebuilt in this area. And it takes about 3 months. Also, if during the breaking of the bones, we have the ends of the bone which are not attached together, some space is between them. In this area between the ends of broken bones, first from the bipotential cells of the inner layer of the periosteum, these bipotential cells will give hyaline cartilage cells instead of bone cells because of the lack of blood vessels. During the breaking down of the bone, during the fracture of the bone, the blood vessels are ruptured, and in the area of the periosteum we have the lack of oxygen, and it causes bipotential cells to develop into hyaline cartilage cells. Hyaline cartilage develops in this region, and it surrounds the area of the ends of the fractured bone, and it helps to immobilize the ends of the bone to ensure their growth towards each other. And it happens here in this picture. So newly formed primary bone tissue combines the ends of the broken bone, and as you remember, always first the reticular fibrous bone tissue is formed, and next it will be replaced with the lamellar bone tissue, and next the cartilage tissue in this area is broken down because it is not needed anymore, and finally healed fracture you can see here. It is covered with the normal periosteum, and the bone consists of lamellar bone tissue. So this is the end of the lecture. Thank you for your attention. See you in class.